In this video we will discuss about some of the results that shows how this capsid is functioning in vitro and in vivo. So before that, let us quickly revise how their system works. So their bacteria that contains the virus that can trigger apoptosis in the cancer cells will enter into the cells, and once it enters into the cells, it will sense the intercellular parts and starts replicating the viral part within this SCV. And after sometimes the HYLE, that is lysis protein, and HYL and E lysis protein will lysate this SCV, and the viral parts will enter into the intercellular parts of the cells, and the replication occurs and the assembly and release of the virus will occurs. This is how the capsid will form. Once it is infected, then only it will start to replicate within this SCV. So what they have designed is that first they will check the in vitro functioning of their capsid using the and they wanted to check whether the full oncolytic virus will be able to reproduce inside the cells or not. So this is how the system is developed. First they transduce the cells only with this long oncolytic virus to see whether they are able to see GFP signals or not. And after sometimes they transduce the cells that have all the three components. In that case, the M-cherry will produced, it will form the polymerase, and this PSSC will form this lytic protein, and the oncolytic virus will be formed, and that will be represented by GFP. So after sometimes when they are able to see the GFP signals, they will transduce the cells with this Wolf's capsid system, and thereafter they monitor the cells. So if the cells has only GFP, then it is the previously transduced this bacteria, and if the cells shows both GFP and M-cherry, it means that it is the their later transduced bacteria. So after 24 hours they are able to see very minimum signals, but after 48 hours and 72 hours they are able to see very like prominent signals. And at 72 hours it is difficult to distinguish that that the cells are been transduced before this capsid system, or the signals are because of the cells are transduced with capsid system. And the temporal projections images is shown at 72 hours. It can be seen here that almost all the cells are transduced or infected with this capsid system. So once they validating the results in the in vitro, they wanted to check the results in vivo. So in that case, what it is, they graft the mice with this H446 lung cancer, and they and after 14 days they starts the treatment. So they did the inject this oncolytic the capsid system in the right hand side of the tumors, and then monitor the signals. And what they are able to find out, 2 days after post infection they are able to see signals on the left hand side, and after 4 days they are able to see signals on the right hand side also, indicating that the viral will replicates on the other sides, on the other tumor sites also. And that can be seen here. And after 14 days they are able to see that in the results are shown in the panel D. So average tumor volumes after 14 days, or the whole 14 days results are shown here. So we can see that the mice that is infected with their capsid system along with the this viral RNA, they shows a decrease in the tumor volume, and whereas the control mice with RPMI or with only the capsid bacteria shows a continuous increase in the volume. And same things they have seen for in the panel D2, where they can see that the volume goes down. The tumor volume goes down in the case where they use the capsid system with the wild type capsid system or wild type SVA with viral RNAs, whereas when they use only capsid bacteria, they do not see any decrease in the volume. I'm not able to distinguish why there are two figures and which one of them represents the which. Maybe one of them is on the left hand side tumors and one of them is on the right hand side tumors. And when they check the mouse survival rate, they can see it really distinguish that in the case where they use capsid system with viral RNA, almost all the mice are able to survive, whereas when they use only RPMI or only the capsid bacteria, the mice dies after the tumor volume reaches to a threshold level. Now they wanted to check these things in the another mice system or actual mice system that is infected with the or that has the tumors. So in that case, and they also wanted to check whether there is any effect of the antibodies against this salmonella or this salmonella virus that is already present within the mice. So whether that has any effect on their tumor cancer diagnostics treatment or not. So what they did is they inject the mice with the bacteria, and after 7 days they engraft the tumors into the mice, and after 14 days they started the treatment and then check the observations. And before they started the treatments, they inject the anti-tumor antibodies to diagnose the mice with the Salmonella bacteria. So what they are able to find out that mice that are already treated with the Salmonella wild type previously are able to show some signals indicating that their capsid system is functioning, whereas the mice that are not infected with the Salmonella bacteria previously are not able to show any signals. This could be because the neutralizing antibodies might be taking care of or might preventing the capsid system to functions, and they are neutralizing the bacteria that is being used in this capsid system. And in terms of the tumor volume, what they are able to see is that only the capsid bacteria where they used and the vaccine treatment with the wild type SVA particles does not able to show a decrease in the tumor volume, but the tumor volume increases in a short duration of time, whereas the vaccine treated mice with the capsid and wild type SVA, that is viral RNAs and capsid bacteria, they are able to show a delayed tumor volume increase, indicating that it is functioning here also, and it is functioning here as per the expectations and helps in delaying the tumor growth rate. And in terms of the survival rate also, they can see that the case in which the mice is vaccine treated and they are used with capsid and wild type SVA, they are able to see an increased mouse survival compared to the case where only capsid bacteria is used or vaccine treated mice with vaccine treated SVA particles or viral RNA particles are directly used. So this results in altogether indicates that the capsid system works better in the in vitro and also in the in vivo, and has a better results compared to the case where direct viral RNA particles are used or direct only the capsid system is used, capsid bacteria is used. And, but this capsid bacteria is just for control, it's not an actual treatment, but this wild type SVA particles can be considered as a treatment, but its efficiency is also low and it's not able to delay the tumor growth in the mice which is complete immunocompetent system. So that's all for this video. I hope you understood how they test the capsid system in vitro and in vivo and what are the results that they come up. If you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.